In this experiment today, we're going to make a model cell, and we're going to look at how the cell membrane works and how the cell membrane can let different molecules through if they're different sizes. We're going to show this by doing the glucose test and the starch test. So first of all, I'm going to show you how to do a glucose test. So I have some glucose solution here. So I'm going to pop some into a test tube. And I'm going to compare this to some water. So we've got plain water in this beaker. And then to test for glucose, we use Benedict's solution. So we're going to put a few drops of Benedict's solution into each test tube. And then to develop the test, we need to use some very hot water. So I've got water from the kettle going in there, and we'll leave that to develop. And then to test for starch, we use iodine solution. So I've got some starch here. We'll pop a little drop on our dimple tray. And again, we're going to test this against plain water. And we use a drop of iodine. So we can see this is a positive test for starch. You can see it's a real blue-black colour. And this is a negative test for starch, this one here. It's the yellow colour of the iodine. So if we look back at our glucose test, you can see this one's starting to change a kind of yellowy green colour. So we'll leave it a little while longer. The other way to test for glucose is to use something called Clini sticks. So I've got some Clini sticks here. The Clini sticks give a little bit more accuracy because they have a scale along the side so we can see the concentration of glucose. So we take one of the Clini sticks, dip it in the glucose solution, and then we can measure it along the side. So the pink is a negative, so if we take it along, we can see the darker the colour, the more concentrated the glucose solution is. So that's probably about that one. So that's positive for glucose. And if we look back at our Benedict solution, we can see the colour now is a real kind of brick red colour. So that's positive for glucose, and this is negative. That's just showing the colour of the Benedicts. Now, next we're going to make a model cell, and we're going to soak it in some water and see if we can get some of our solutions to pass through the cell membrane. So to make a model cell, we're going to use something called whisking tubing. So I've got some tubing here. Now this, like a cell, has got a semi-permeable membrane, so some molecules are able to pass through it. So I'm going to soak it in some water. And I'm then going to tie off the end of the tubing to seal at one end. And we're going to fill it up with starch and glucose solution. So we've got it tied at one end here. I'm going to put half glucose and half starch in here. I'm then going to tie it up and cut it. And there we've got our model cell. So it's tied at both ends, sealed at both ends, and inside we've got our starch and glucose solution. So I'm going to pop it into the water, and we're going to soak this for 30 minutes. And then we're going to test the solution to see if anything has been able to pass through the membrane. So this cell here, this one has been soaking for 30 minutes. You can see our model cell in there. So we're going to test this solution. So first of all, we'll test it for glucose. So I'm going to take some of the solution, pop it in our test tube. A few drops of Benedict, and again the very hot water is needed. And we'll leave that one to develop. We'll then test our solution for starch. So we can see we're getting a negative result for starch. So this means no starch molecules have been able to leave the model cell and enter the water. So the starch molecules are too big to go through the semi-permeable membrane. If we look back at our glucose test, just starting to get a colour change. I'll leave that to develop a bit longer. And we'll test our solution with one of the Clini sticks. So if we take one of the Clini sticks, dip it in the solution here, and then the pink is a negative result, and then as it gets darker it's positive. So we can see we've got a positive result for glucose, and it's about the third one this time. So if we look back at our Benedict solution, you can see it's just starting to go that yellowy green colour. So we'll leave it a little bit longer. So we're getting a positive result for the glucose. So this means the glucose molecules are a bit smaller and are able to pass through the semi-permeable membrane of the model cell. So you can see we've now developed that brick red kind of colour, the orangey red colour, so that's positive for glucose.